Rong started his training at the University of Science and Technology of China, where he earned a bachelor's degree, and then he moved to the US to obtain a PhD degree at the University of California, Berkeley. He is today an endowed professor at Yale University, Harold Hutchinson Professor of Biomedical Engineering, and he also has an appointment in pathology. Rong's research is really interesting, and it has been centered around the development and also implementation of single-cell and spatial omics technologies that are studied in the context of human tissue function, both in normal physiology, including aging, and in disease. He's the one who developed single-cell 42-plex cytokine assay, and this is up to date really the highest number multiplex for single-cell protein secretion analysis. He also was the one to demonstrate for the first time the spatial multi-omics sequencing by developing deterministic barcoding in tissues that was used for mapping whole transcriptome and about 300 proteins pixel by pixel directly on a fixed tissue sections. I mean, we all understand how helpful and important this is. More recently, he extended this approach to spatial profiling of all kinds of RNA and also of single nucleotides. His research also has led to the spatial epigenomics. So it's a very like a broad, comprehensive field of research, which does have a common ground, as we will hear today. Dr. Fenn is very broadly published and very highly cited. He published multiple articles in each of the top journals, Cell, Science, Nature, Nanoletters, and the impact of his work is seen in many different ways, including his excellent citation record. So he received multiple awards throughout his career, and I will just mention a few. He, some years ago, as a more junior faculty, he obtained the Bill and Melinda Gates Foundation grant from the cluster of grants that were about grand challenges exploration. He's also Kavli Fellow, which is a prestigious appointment. He's a fellow of the American Institute for Medical and Biological Engineering since 2019, and a fellow of the National Academy of Inventors since 2021. And most recently, last year, he obtained the Momentum Award, which is given by the Biomedical Engineering Society. Rong will be speaking to us about ushering in a new era of human tissue biology via novel single-cell and spatial multi-omics. Rong, thank you so much for being with us, and you can now take over the screen.
Oh, thank you so much, Gordana, for the very kind of introduction. It's really my honor to come to speak at your Tissue Talks seminar series. And so I recently started to talk about this concept of whether or not with the new tools that started to emerge over a couple of years, we're really at a crossroad, kind of getting to a whole new era of human tissue biology. It's a little bit proactive title, but I'm happy to hear any comments, feedback from the audience today. And all right, so I'm gonna just dive right in since I have so many slides. This is my disclaimer. I'm reading some of the history about tissue biology. I found fascinating. Actually, 5,000 years ago, Egyptians already started to look into human tissues, organs, and not something just so obvious like a brain. The actual owner major organs, the terminology, the nomenclature actually came from Egyptians back to 5,000 years ago. That was amazing. But they also already recognize some detailed tissue features, for example, meninges, and something so amazing like a cerebral spinal fluid. So hard to understand even nowadays. But 5,000 years ago, Egyptians sort of look into that. But how to understand that like a tissue physiology, biology back to that time, just look into human body, right? So dissection, do dissection and look into human organs and tissues, and that tradition retain until kind of later stage of the Greek and the Roman ages, and the dissection is no longer kind of allowed or somewhat controversial. And so another important figure in the history of medicine, Hippocrates, is regarded as the father of medicine. And if you're a medical professional, you'll probably kind of start your career by reading out this Hippocratic oath. And so what he already emphasized back to 2,000, 3,000 years ago was you really want to understand human tissue biology back to that time, which is sort of anatomy, you have to look into the form of the human body, right? So when you really want to understand human tissue biology, the way to go is to actually examine the human organs and the human tissues. All right. And over the past kind of century, in particular, past 60 years, and molecular biology, since the discovery of the DNA double helix and the whole central dogma of molecular biology, that completely changed how we understand biology. So no longer just a top down from the whole body to organ to tissue, you really now understand the biology from bottom up. Okay, every single molecule, and from the DNA, the genetic blueprint to which genes are expressed, which genes are not, and which genes are going to give rise to proteins to eventually dictate the molecular biological functions. But somehow there's a divide between those two fields, like a molecular biology and a human tissue physiology, and that to some degree, it kind of defines whether or not you're more basic science or you're more clinical physiology. But I have been thinking why we cannot understand the detailed molecular cellular biology in the human physiological systems level and cell by cell, and and across the entire large area of the tissue and even the entire organ, right? So if you look into the human tissues, this is a review article written by Professor Joanna Joyce back to 2005. When I started my postdoc 2006, I read this article. I was very fascinated by what she articulated, the concept of the tumor microenvironment. This is a tumor tissue, but normal tissue is also pretty complex. And in the tumor tissue, that's the first time I realized tumor is not just a bag of tumor cells. There are so many different cell types in the tumor microenvironment. And also the cells are not just sitting there in complete random fashion. Actually, they form very complex spatial tissue architecture, right? So I was kind of thinking whether or not we can really map down this cell by cell and also every cell understand molecular biology, and then you can reconstruct this underlying molecular cellular mechanism, but in a single cell and a subcellular level to really understand how every single sort of cell works in a complex tissue microenvironment, such that we know a lot better how to do targeted treatment, how we do much more accurate and personalized diagnostics. And so that has been kind of my dream since, wow, time flies, almost 20 years ago. And but back to that time, literally, if you want to look at single cells at a reasonable high throughput, but also a large number of biomolecules, that was not possible at all. That I kind of view that as really the ice age of the single cell omics. And one of my friends, Bo Huang, he was graduate student with Dick Zier at Stanford. he devised this microfluidic system to entrap a single cell here in the lights, and you perform some chromatography, gel electrophoresis, and you can count one or two types of proteins, and that's a science paper, right? But if you really want to understand the complexity, heterogeneity of different cell populations in a complex tissue like a tumor microenvironment, well, way far from there. If you take this device to look into tumor heterogeneity. And so what I proposed back to that time focused on two things. One is, can we get a relative higher throughput, hundreds of single cells or even 1,000, but each cell you can look at a pan of tens of different physiologically relevant proteins. For example, cytokines produced by T cells to mediate their cell-cell communication. All tumor cells can also produce cytokines, chemokines to hijack maybe non-malignant cells or kind of hijack the immune cells to do something bad together. So I developed this device back to 2006, 2007 to measure single cell secreted proteins. And so now that technology has been kind of fully commercialized by Bruker. And in the meantime, we over the past 10 years, we also started to develop our own single cell gene expression analysis tool, aiming for something like a fully automated sample in and the library out. And you can have sort of fully automated process and also high throughput process to look at a large number of single cells as required for dissecting the complexity of the tumor microenvironment. So we applied those tools. We developed and also commercialized single cell cytometry to study, in this case, the pre-infusion CAR T cells from two clinical trials started Penn about 10 years ago. Those are the first two CAR T cell immunotherapy clinical trials, such that we can look into the long-term response between the individual patients and this single cell profile, functional profile in particular, in this case, the antigen-specific CAR T cell activation states. So what we found quite striking and very counterintuitive. So we found if you study T cell biology, if you want to make the tumor, the T cells are very effective in so anti-tumor response. You want to increase their gamma interferon production, TF, and granzyme B and porphyrin. Those are kind of classical type 1 anti-tumor response. But what we found, all the samples demonstrate pretty good type 1 response. But very subset of the cells producing type 2 cytokines turns out to be strongly correlated with long-term response. And so long story short, I think this is kind of very counterintuitive. So you really need a very small fraction of those type 2 cytokine producing CAR T cells to maintain the whole population in a homeostatic state such that they don't become completely exhausted. And you have the perfect balance between the type 1 and the type 2 to give rise to long-term remission in those patients. It's a beautiful work. We also figured out the mechanism because those type 2 cytokine producing cells really regulate the dysfunctional TIM3 positive exhausted T cells. So a nice story we can figure out from large scale single cell sequencing, large single cell multi-omics data. And so I think in the past, people always say omics data or single cell omics data is very valuable for hypothesis generation. But now the data is so rich and very high resolution and actually can directly learn biology from your data itself. Um, but this is one of the very few cases. Okay, the work was published a couple months ago. Actually, something very similar was discovered by my friend Li Tang at EPFL, and so there's kind of two papers back to back published in Nature. But back to this diagram, right? So very few cases in a field of cancer immunotherapy research, you just look at single cells, that's good enough. But the actual function of individual T cells in the tumor, you have to put in the context of the spatial tissue architecture, right? So now I think people realize that that critical need, that person need about 15 years ago, and started to develop this now so-called spatial omics technologies. Uh, I would say that the field is very broad, and but spatial transcriptomics or spatial gene expression remains the mainstay in the spatial omics technology field. And this spatial transcriptomics was chosen by Nature Methods as the method of year 2020, and Professor Xiaowei Zhang and Professor Joachim Lomberg wrote two perspective articles to kind of review the progress of the field in two distinct directions. One is imaging-based spatial gene expression profiling, and second is sequencing-based spatial gene expression profiling, direct in tissue spatial resolve, right? We're probably the first group that realized, oh, that's not enough because which genes are expressed, which genes are not. That tells you the cell type, but what drives, what controls, what regulates the gene expression. So you need to understand across the whole central dogma of molecular biology. So we basically demonstrate the first the spatial multi-omic sequencing and later further expand to spatial epigenome sequencing such that you can understand the mechanism controlling gene expression rather than just the gene expression alone. And in recent years, I think genome proteome-wide spatial proteomic profiling started to emerge, and also our own interest beyond the central dogma molecular biology from DNA to RNA to proteins. Can you really understand the function as well? For example, metabolic activity. So in collaboration with my friend Professor Lin Yanxi at UC San Diego, we are combining our spatial omics profiling and her advanced optics tool called stimulated Raman microscopy, stimulated Raman scattering microscopy to link cell by cell the metabolic activity to the underlying cell types or the mechanisms profiled by spatial multi-omics. All right, to put in a roadmap of the NGS-based spatial multiomics or spatial omics, where we made our own unique contribution to. And the landmark paper was published by Professor Joachim Lamberg 2016, using the DNA microarray to capture messenger RNAs released from the tissue slide you place on top of the DNA microarray. You permeabilize the tissue, you fix and permeabilize the tissue, and the DNA RNA molecules diffuse out of the tissue and then captured by the DNA microarray spots. Every spot has unique DNA barcodes, and then you basically incorporate the spatial barcodes or spatial address codes into the cDNA when you synthesize the cDNA using reverse transcription directly on the slide, on the glass slide. So that's how it works. Over the past couple of years, I think many other technologies were reported, but they are more or less along the same line, based on the same fundamental principle. But what we came up with is kind of opposite. Instead of capturing messenger RNAs, we send the spatial zip codes into the tissue to tag either RNA or tag the proteins or even epigenetic mechanism. For example, where the chromatin is more open, where that's kind of chemically modified by those histone marks. So you learn a lot of biology. You can directly tag those biology spatially, and after your sequencing using NGS, you can spatially construct the biological mechanism, not just a gene expression. All right. So a number of papers were published over the past years to demonstrate a set of tools to spatially map the protein RNA and the epigenetic, or when you combine them, spatial multi-omics sequencing, which is literally regarded as the holy grail of the spatial omics field, because now you see which genes are expressed, but you also know what underlying mechanisms are controlling the expression of those genes. All right. So as I said, nature methods highlight spatial transcriptomics as the method here. Our work was also chosen in the same issue to emphasize starting now, multi-omic sequencing goes spatial. And a couple of months ago, is another exciting news in the whole field of spatial biology, spatial omics is spatial proteomics was chosen as the method of the year. I was honored to write this perspective in this issue, emphasizing the study of spatial protein profile needs to be put in the context of the central dogma of molecular biology by doing this spatial multi-omics, including proteins, such that you know which proteins are expressed or not, but you also know what that does to either controlling the enzymatic activity associated with RNA or epigenetics or the metabolic function. All right. So as I mentioned, Professor Xiaowei Zhang and Professor Joachim Lomberg wrote those two perspective articles highlighting two distinct approaches, imaging or sequencing-based spatial omics. Each of those has its own pros and cons. Then I was asking myself and my laboratory, so can we somehow combine both on the same tissue slice as they get the best out of the both worlds? And so we decided to demonstrate this by combining high-plex immunofluorescent imaging, such as this assay called a Codex developed by Professor Gary Nolan at Stanford. And we image 53 protein markers in this human lymphoid tissue, lymph nodes, or lymphoma. But in the same tissue, we're able to perform spatial omic sequencing, either transcriptome or epigenome. So in the whole field of spatial transcriptomics in the past, if you do this type of sequencing, after you perform unsupervised clustering to identify all tissue features, each spot you can do a little bit further analysis by integrating your spots with single cell reference data, such that every spot you can generate a pie chart to tell people, oh, what's the percentage of different cell types in every single spot. But in our case, you can do much more accurate decomposition because every single spot you have the high-plex immunofluorescent imaging data, you know exactly what cells you have. So that's no longer pie chart. You actually can get a single cell resolved spatial transcriptome using this high-plex imaging cell typing data to guide the deconvolution or decomposition of spatial transcriptome into individual cells, creating this full single cell resolved spatial spatial kind of genome-wide transcriptome atlas or genome-wide spatial epigenome atlas. So now we're applying this tool to two different consortium projects. One is called the Cellular Senescence Research Network, called the CENA, in which we study how human lymphoid organs, how our immune organs gradually change during aging. There's a spatial temporal process associated with cellular senescence development, but directly in the human tissue. And another related areas in malignancy, right? So if the lymph node started to develop some malignant cells and the eventual full-blown lymphoma, what's that spatial temporal atlas you can map out to understand this lymphoma. All right. And I also mentioned beyond the central dogma of molecular biology, DNA, RNA to proteins, can you understand some molecular or biochemical function, right? Such as metabolism. What you see here is called a redox activity, corresponding to the relative ratio between glycolysis versus oxidative phosphorylation using the advanced microscopy technique developed by Professor Lin Yanxi. we see individual cells, which is more glycolytically active, which is more reliant on oxidative phosphorylation. But not every cell you see here, because we perform the spatial codex imaging, hyperplex immunofluorescent imaging, we know exactly what that cell is. Now we can do cell type specific metabolic analysis, and also in the same tissue, we can do spatial transcriptome. Then you can really link the genome-wide transcriptional program to cell type and then to metabolic function. All right, so we also apply the same approach to look into lymphomagenesis from normal to early indolent follicular lymphoma and to more aggressive diffuse large B-cell lymphoma. And it's a very interesting findings, a lot of interesting findings from this fully integrated analysis. To show you one of those. For example, this redox glycolysis versus oxphos, that's one of the kind of energy pathways in the cells, and the others, for example, the lipid metabolism, right? So it turns out in normal lymph nodes, most B cells in either germinal center or in other regions, they just require one of those to drive or to provide energy supply for those kind of normal non-malignant immune cells. But somehow in lymphoma, the B cells is really applicable to both metabolic pathways. To some degree, that makes perfect sense, but now you actually see that cell by cell. I still feel that's mind-blowing. Even though we know tumor cells are require more energy to drive their sort of high metabolic activity, but also drive their proliferation and migration. So, but still now you see which ones. I feel that's still pretty mind-blowing. Um, so now, okay, back to spatial transcriptomics. I was often invited to speak about spatial transcriptomics, but I said, okay, maybe I'm not the right person to talk about it, because from the very beginning, I found that boring. If you just look at gene expression, starting from 10 years ago, spatial transcriptomics using imaging and sequencing were demonstrated. And until now, the 10 years now, if you look at what spatial transcriptomics can give you, I have my own understanding, but still go to ask a ChatGPT or Gemini, those chatbots. And the key messages are very consistent. You look at gene expression by measuring RNA, but typically just a messenger RNA. I think from either of those chatbots, you get the same type of answer, right? And however, and the every RNA molecule, which you learn from your molecular biology class, is very dynamic. Okay, starting from the nascent RNA right off the transcription, and then go through some splicing, some editing process to give you mature RNA, and then some mature messenger RNA is going to be transcribed to give proteins, and eventually they're done with their job. They're going to be degraded. This is a dynamic life cycle of RNA molecule is actually regulated by many other non-coding RNAs, and non-coding RNAs are kind of comprising 95, even more than 95% of total RNA molecules. But current spatial transcriptomics technologies, most of them don't tell you anything about the dynamic life cycle of an RNA. They don't tell you anything about this kind of non-coding RNA. They just pretty much teach you which genes are expressed, which genes are not. So I really think you'll miss majority of the rich RNA biology using current spatial transcriptomics. And also another kind of challenge, or another for us, I feel present need is to understand the rich RNA biology. Ideally, if you want to study that in the context of human tissue biology, you need to look into human organs and tissue, as sort of pointed out by the father of medicine, the Greek physician Hippocrates. And the human tissues, it's a very interesting article, I think like a year ago in Science. I found that's fascinating. We always blame, okay, human tissue, whatever tissue specimens, easy to degrade, And formalin turns out to be a magic. You can fix the tissue in formalin, but afterwards you really cannot do much. And it's a very interesting sample digged out in Florida, and turns out that's a 8,000 years old kind of Asian human skeleton in the skull. They found still like a jello-ish brain, and there's and some RNA DNA molecules in there still remain intact and do some molecular analysis. I have no idea how that brain tissue was preserved, what's the magic chemical, but in the current modern medical practice or pathology practice, we use formalin to fix those tissue specimens. If you look at this human brain bank, this is a brain bank at Harvard Medical School, one of the largest brain bank in the world. It's a lot of brains stored in room temperature but fully fixed in formalin. In clinical diagnostic tissue specimen, and most of them are formalin fixed and then afterwards embedded in a wax block called a FFP tissue blocks that preserves the tissue morphology very well, and that's the gold standard nowadays still for pathologists to perform their diagnostic testing, and that can tell you by just looking at the morphology of the tissue and the cells, and to give the diagnostic, to give the diagnosis. And but I think people constantly blame the formalin after you fix, and then particularly in the paraffin embedded format, you really cannot do much molecular biology. You can probably extract the RNA, some sequencing at a bulk level. You can extract the DNA to look at kind of bulk level, but there's no way to really understand cell by cell the rich molecular biology. Recent advance in the single cell resolution imaging and or phage, you can now see gene expression, but beyond the gene expression, you still don't learn that much. So we really want to have the molecular biology, okay, the rich information of molecular biology stack on top of the tissue histology image to help our superheroes. They are really my superheroes, our pathologists, and give the diagnosis, and also kind of desperate require more superpowers such as those molecular information to do better diagnosis and patient stratification for personalized treatment. And so now we're asking, can we kind of obtain the rich molecular biology information from ideal the paraffin embedded, formalin fixed paraffin embedded clinical archival tissue specimens? That has been a project we started about over 2 years ago. And so what I want to tell you is that work, and the work that's surprisingly well. It's actually uh, it's a total surprise. We could not anticipate that this actually worked out even better than the fresh frozen tissue samples, and worked out better than the standard fish. Those kind of supposed to be much easier and much more reliable protocol, but actually the paraffin tissue samples can give you even better data and more molecular biology information. Okay, the trick we play with was uh, first proposed and implemented by Professor Steve Quake a couple years ago to do single cell total RNA sequencing. And that's not a high throughput, but use kind of individual PCR where you put a cell there, kind of relatively low throughput total RNA processing. But how come you can do total RNA? The trick he uses is you use polyadenylation enzyme to add a polya tail to all kinds of RNA molecules, not just the messenger RNA, which already had the polya tail. But all the non-coding RNAs, they don't have polya tail. Now you add a polya tails to those RNA species. So now we're asking, can we do the same chemistry in a tissue without tissue digestion dissociation? Can we add a poly-A tail to all different RNA species, and even the RNA molecules highly fragmented? We can add our poly-A tail to all those individual fragments. Turns out that's great because in FIP, people always blame RA degradation, RA quality is not good, but we found actually not really, the RA quality is not bad, but they are very fragmented. So now once you polyadenylate, you can polyadenylate all those fragments. Turns out you get a much better, much more rates for every single gene. You can have much better sensitivity. All right. So, but what is super cool is now just you detect you have better sensitivity, which I'm very happy about. But we can detect all different RA species, kind of microRNA and the tRNA, snow RNA, and the and the ribosomal RNA, and for every single tRNA, actually you can see every single amino acid associated tRNA. So it turns out the tRNA is the first non-coding RA that was recognized by Nobel Prize. And okay, the joke I often tell is I used to drink a lot of Red Bull. Okay, of course, a lot of caffeine in there, and but they also put a lot of taurine in Red Bull. And so I was always wondering when I drink too much taurine, where the taurine goes, right? So now with our technology, you can say taurine associated TRA, and then you know where the taurine goes if you drink too much Red Bull. And all right, as I said, we have very high quality data using this kind of pathology compatible FFPE tissue spatial transcriptome. If you look at just gene count and UMI count, we can do even better than fresh frozen sample spatial transcriptome and also the probe-based spatial transcriptome. I thought it's no way to do better than just a probe-based, fish-based, and now actually this even could be even better. Uh, so as I said, all the different fragments we polyadenylate. Now we get a full coverage of every single gene, potentially can map down all different splicing isoforms rather than just a gene expression. Since this is down spatial, you can map out the spatial region-specific splicing isoform across, for example, in this case, mouse brain or different anatomic regions. You can quantify different splicing isoforms. For example, one of those we saw the expression, this protein phosphatase 3CA subunit, expression everywhere in the dentigerous isocortex and CA1, but this specific skipping isoform, you see the skipping isoform only in the dentigerous. All right, if you don't have the splicing isoform resolvable spatial transcriptome, you cannot discover that interesting thing. You can also see something very difficult to map out even at a bulk level, like A to I RNA editing. Now you can still map out spot by spot across the entire tissue. All right, so the real value of this tool lies in the mapping of clinical archival tissue specimens, and we'll use this specific case, the clinical case, to illustrate the power of this approach. This patient had a stomach upset for like 2 months and came to Yale, and you have a hospital and an endoscopy and a 2 biopsy collected from the stomach. And this is the one, this is the second, and my pathology colleague, my friend Mina Su, given the diagnosis. The biopsy 1 is kind of relatively lower grade lymphoma, and the biopsy 2 has some manifestation of large B cell, and likely it's a large B cell lymphoma. She also requests order a lot of molecular testing. So altogether, this is now, it's not cheap at all. It's a lot of molecular tests to really nail down the diagnosis. And this patient actually has this transformed DLB cell transformed from low-grade MALT called a mucosa-associated lymphoid tissue. And if that happens, that doesn't happen very often, but if that happens, the patient prognosis is very poor, and how to treat that patient, the chemo is still in the mainstay, and but some patients may respond to the targeted therapy, PI3K inhibitor treatment, but we really don't know who's going to respond, who's going to. It's a no way still now, no biomarker exists to tell you. And so now we perform spatial total RNA transcriptome sequencing called a pathodibbit. You get a very nice clusters and look at different cell-specific signatures. Those are the B cells, and basically those are malignant B cells. And you also see some kind of malignant B cell signature in the kind of non-malignant region. You see basically tumor infiltration into those connective tissue as well. What is super powerful nowadays with the latest breakthrough in machine learning AI is the computational pipeline developed by Professor Miyali at Penn allows you to kind of integrate this high resolution tissue histology image with spatial transcriptome to derive almost single cell resolution spatial gene expression profile, and you can even infer the transcriptional profile in a region not covered by our spatial mapping to give rise to this whole tissue section scale spatial protein or spatial transcriptomic profile. With that, you can do your own annotation to identify a lot more different cell types. I just show you majority of those. You can even kind of do subclustering to identify so subsets of the different cell types, in particular the tumor cell types. So now you see the tumor region, you already see a lot of fine features based on the transcriptional profile. I think the tissue histology really cannot tell you. And it's so hard to identify so many different fine features. In particular, right here is those lymphoma, you see many more different kind of molecular details. And so, as I said, the same patient, we also had a high-grade lymphoma, and this high-grade lymphoma, we performed the same analysis that gave rise to this almost single cell resolution spatial transcriptome or transcriptomic atlas by applying this I-star AI machine learning algorithm. But every single cell you see here, you have transcriptome-wide unbiased the genome-wide gene expression profile. Now you can ask a lot of interesting questions. For example, we notice a group of macrophage cells certain distance from the tumor zone, and here the macrophages are really close in the vicinity of the tumor, or even infiltrating into the tumor. We're asking this, they're in the same macrophage or no. So now can perform this differential analysis between the macrophage here and there. We saw, oh, in the large B cell lymphoma, you do see a lot more type 2 like macrophage, M2 macrophage, and you can see many other molecular pathways that are either upregulated or downregulated in this more aggressive diffuse B cell large lymphoma region for those macrophages. So if you learn so much more molecular biology in this case, rather than just in the cell types, right? If you do high-plex immunofluorescence imaging, you only know cell types. You cannot ask more questions about the molecular pathways. Speaking about cancer, you always need to kind of understand the underlying genomic roots or genomic alterations driving the development of different tumor clones. If you're able to do so, and that's kind of like a tumor mutational burden if you can map down all the mutations, right? But we're able to map down the whole transcriptome wide and also the full length transcripts in this case. We potentially can map down all those mutations as well. I told my postdoc, can we give it a try? Turns out that worked out well, and on a single nucleotide variants, if you look at the high SNV count region versus low, that clearly correspond to the tumor region versus the non-tumor benign region. If you know the clinical marker for tumors, such as BCL2 in this case, you do see very strong concordance. But if you don't know, this should be your ground truth, because the cancer to some degree is still like a genetic or epigenetic disease. So if you see any changes in aberrant changes in the genome, that serve as the ground truth to really diagnose the sort of cancerous cells, right? But since in this case we have genome-wide profile, we can further ask the questions like, can we further kind of subcluster or subclassify the tumor cells into different subclones. Yes, we did that, and we further generate, in this case, the mutational profile, SNV profile based classification of tumor subclones to construct the tumor clonal phylogenetic tree. People have done this using single cell analysis, but in this case, you see where they are, all different subclones, you see exactly where they are. So this kind of spatially resolve the tumor clonal dynamics. And so you can also see individual mutations, individual SNVs. One of those got our attention, and later we search literature. Wow, this mutation have been studied by R.A. Melnick, and it's a I think an expert in B-cell lymphoma research. So he found this mutation can drive the formation of memory phenotype, and as a result, that leads to more aggressive lymphoma and potential also more resistant lymphoma. And so we also combine the copy number variation and the single nucleotide variation to selectively define the tumor clonal dynamics. And so altogether, you can really understand this kind of genomic alteration driving the clonal evolution of the tumor, the same patient, all right? The same patient, what's the clonal evolution of the tumor in that particular tumor in that particular patient, right? All right, so as I mentioned, I know I'm running a little bit out of time, but the non-coding RNA, as I mentioned, that's more than 95% of the total number of RNA. And that constitute very rich RNA biology, it turns out, because they're playing very important role in regulating the messenger RNA and regulating the post-transcriptional gene activity. And so now we can map down all of those, and from the same tissue, the mild lymphoma. And just to show you one example of microRNA, that's the most difficult RNA to detect, no matter single cell or spatial or even a bulk. And from FFP, that has not been possible to detect the microRNA. Again, the microRNA is another Nobel Prize winning non-coding RNA, non-coding small RNA discovery. And from that same lymphoma tissue, we detect over 1,800 microRNA, and total number of microRNAs in human cells about 2,000. We detect almost entire pool of microRNAs in our sample. So now you can do all kinds of interesting analysis, for example, between tumor and a non-tumor, or between different regions of the tumors. You can do differential analysis on microRNA expression. In this case, what I'm showing you is differential microRNA expression between the malignant tumor versus non-malignant region, right? And so you can also look into, so in this case, I show you two cases. One is mirror 121, it's a known uncle mirror. Another is 155. We don't know what happens to that mirror, but let's look into the biology. So in the microRNA field, there's a lot of computational methods out there that can predict the genes upstream of each microRNA or downstream, sort of the target gene of the microRNA. But now we have the data actually from every single spot. You see the predicted genes upstream or downstream, which ones are actually detected in our data. And you look into the ones we actually detected and then put in the gene ontology analysis, say, oh, which pathways are upregulated or downregulated. So now we see upstream of mirror for the 5, the alpha kappa B signaling is upregulated. And downstream, that's the PI3K AKT. Now the biology becomes crystal clear. Okay, kind of somehow persistent inflammatory response in this region leads to alpha kappa B activation, and that activates the expression of mirror 155, and mirror 155 triggers an activation of PI3K AKT. That's a pro-tumor survival and a proliferation pathway. So now the biology pretty clear. I mentioned a little bit of some of the patients may respond to PI3K inhibitor therapy, but we don't know who gonna respond. So now from this data, this patient are probably gonna respond because you see PI3K AKT pathway activated and activated through miR-155. And if we have therapeutic targeting strategy to inhibit miR-155, that could be another therapeutic approach. But if not, miR-155 could be a biomarker to predict who's going to respond or who's going to not, right? So now just a single case, you just learn so much RNA biology, and what you learn potentially can direct tell you how to better manage the disease in this patient, how to better treat this patient, right? So this work has been published and also chosen on a cover of that issue focusing on molecular biology. I was, oh, why they choose our paper to be on the cover? There's a molecular biology. Now I said, okay, now we really learn so much molecular biology from this peripheral block clinical archive tissue specimens. That's gonna really change how we study human tissue biology in the future. Just to show you, we also can do the same thing for epigenomics. I don't have time to talk about epigenomics, but just let you know, I think a very recent development in my lab over the past half years really unlocked an opportunity to look into epigenetics as well in clinical archival tissue specimens. All right, next a couple of slides, kind of maybe a little proactive, and what the future of human biology, human tissue biology research and drug discovery gonna look like. I don't exactly know, but I think we have whole new opportunities out there. And so one of my colleagues, very esteemed professor at Yale, is, oh, mice get models, human get diseases. And so mouse models cannot fully recapture human diseases, too complex indeed. And the human clinical trials are actually the best experiments. But most of the clinical trials and afterwards, you get a lot of perfect blocks, you don't learn that much. And clinical trials are somewhat flying blind. And afterwards, you even cannot retrospectively learn very rich molecular biology from them. But I would say that's changing. And some shocking news, I think earlier this month, and the FDA even decided to kind of phase out gradually the animal testing required for some of the drugs, in particular monoclonal antibodies. And so in parallel with sort of the molecular biology we can learn from the human clinical trials and the paraffin blocks, another way to model human biology is like a human organoids, and you can even integrate organoids in much more well-controlled system. For example, like organ on chip, Gordana is really a pioneer in the field, and NIH and NCI have their own initiative to drive this kind of tissue engineering modeling microphysiological system to understand human physiology, human biology, right? And so pharma also realized, wow, that's whole new opportunities out there, and we should embrace. And Roche launched their whole institute called the Institute of Human Biology. One of the main focuses there is to develop organotypic tissue models, human organotypic tissue models. Within the same institute, they also have very advanced spatial omics mapping technique. Now combine both, you are really at a cutting edge, you are really at a kind of this beginning of the new era to understand human biology. I would like to use this slide from my own study to summarize. I think we're really at the beginning of this new era, and from the clinical biopsy tissue in the past, you don't learn that much, at least cell by cell. Any of those molecular information is so hard to figure out. Now you almost have everything, at least an RNA level, all kinds of RNA, and including TCR, BCR. I didn't talk about. You can actually track the TCR, BCR clonal dynamics, and also rich epigenetics. And using all the AI machine learning, you can integrate everything together. And the recent development in CZI and ARC and many other consortium that build a molecular biology-based virtual cell. I would say this data is the way to connect a virtual cell to human. And so we are really at the beginning of new era to understand human biology much better. All right, so I'm running out of time. I would like to thank all the current and former lab members. Without their devotion to science, I'm not going to be here to speak to you. And funding support from NIH and some foundations. And thank you very much for attention. If time permits, I'm happy to answer a couple of short questions.
Wrong. Okay, this was really amazing. So your science is absolutely awesome. So let me just for 1 second introduce our next speaker next Wednesday. So we will have Chris Chen, a very different talk, but also something very important for the field. So he will talk about how to build tissue function through engineering and with focus on the forces within. So we really ran out of time, but maybe you can take like 1 or 2 questions. One comes from Gilad, London. Fantastic. Can you perform enzymatic experiments on slides, either fresh or embedded, followed by in situ sequencing?
Yeah, this is a great question. Some kind of live cell, live tissue experiments can be done on brain slices or even lung slices. I think here at Yale, my colleague, that's durable, kind of ex vivo tissue slices. And afterwards, okay, you still have to fix that becomes like a single snapshot. But I want to say the snapshots are not truly static. You can either record a lot of for example, those CRISPR recorders. And for metabolic activity, you can use some metabolic tracers to record. So a lot of recording mechanisms. And also throughout that process, right, if you perform your enzymatic live cell, live tissue assay, and during that process, what's the turnover of those different RNA molecules? And so turns out we can computationally trace back. Uh, so I just want to say the single snapshot is not 100% static, and actually a lot of temporal information recorded over there, and also that can be done after you perform your live cell or live tissue experiments. I think in my lab, I think one of my postdoc was doing kind of live brain slice or organoid calcium imaging recording, and afterwards we do the spatial profiling.
Okay, and if you could take just 1 question that is sort of ties into what you just addressed, is from Ophelia Bagdasarayan. Thank you very much for this incredible talk. How long is the turnaround the time for patient diagnostics with the omics data incorporation?
Yeah, I think cancer, most cancer diagnosis, I think initial diagnosis, that's a morphology-based H&E. Okay, that will not change. My best friend Mina Sue, she said, okay, I, I was giving only literally a few seconds to do the first quick look, and if that's now a very tricky case, I'm gonna give diagnosis. But afterwards, right? So if you want to know how to treat that patients, and cancer, to most of the cancers, that's really kind of somewhat like a chronic disease. It is okay to take a few weeks to gather rich molecular biology information to tell the doctors how to better manage that patient, right?
Okay, we need to let you go. Thanks
Mhm.
again. Thanks very, very much. This was fantastic. And hope to see everyone next week for Christian's talk.
Thank you, Gordon. Thank you, everyone. You have a wonderful evening.
You too.
Mm.